Hello. I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into medical school and other professional programs. Today, we're talking about another fundamental concept in biochemistry and cell biology on the MCAT, ATP production. Specifically, where does it come from and where does it go? This molecule is the energy backbone of virtually all cellular activities, from muscle contraction to nerve impulse propagation. Understanding how ATP is produced is crucial for any aspiring medical professional. At the end of the video, we'll go over some common questions on the MCAT that you will likely see on the exam. So give them your best shot. First, what is ATP? Well, ATP, or adenosine triphosphate, 3 phosphates, it's a complex organic chemical that provides energy to drive many processes in living cells, like muscle contraction, nerve impulse propagation, and chemical synthesis. When ATP is broken down into ADP and an inorganic phosphate, energy is released to be used by the cell. And let's take a look at what's going on just so it's very clear. ATP, the adenosine, is this guy up here, or an adenine. You might have seen this on a nucleic acid as a base. Then we have a ribose, which is just a sugar. And then we have 3 phosphates. Tri means 3. Phosphate is this guy. So we have 1, 2, 3 phosphates all linked together. When we break this down from triphosphate to diphosphate, or 2-phosphate, we're going to lose one of those phosphates. Now let's talk about the pathways that are making ATP and where it's going. ATP is primarily produced through cellular respiration, a multi-step process during glycolysis, involving glycolysis, the Krebs cycle, and most importantly, oxidative phosphorylation. Glycolysis, which occurs in the cytosol, breaks down glucose into pyruvate. This generates a small amount of ATP, 4 total ATP, but it took 2 to start it, so you get a net of 2 ATP just from glycolysis here. The Krebs cycle next will take that acetyl-CoA to produce even more ATP. You'll notice I've also circled where we're making NADPH and NADH, and FADH2, excuse me, not NADH. You'll also see that I've circled where we're making NADH and FADH2. This is important because these will go into the electron transport chain, but we'll get to that in a second. The real powerhouse is the last stage, oxidative phosphorylation, occurring in the inner mitochondrial membrane. Here is where the majority of ATP is produced, and that's where we're going to focus on next. Oxidative phosphorylation is a complex dance of molecules and reactions, primarily involving the electron transport chain and ATP synthase. The electron transport chain, or ETC, is a series of protein complexes that pass electrons from donors like NADH and FADH2 that we talked about earlier, and actually turns it into something useful. Ultimately, energy, ATP. But how does it do this? Well, NADH and FADH2 are going to pass electrons through the chain until they are ultimately accepted by water, by oxygen to produce water. While it's doing this, the energy is being used to pump hydrogens from the matrix of the mitochondrial into the intermembrane space. This is going to cause a buildup of hydrogens outside the space, also known as the proton motive force. This proton motive force will move through a turbine called ATP synthase to produce ATP from an ADP to ATP reaction. This is tightly regulated by the cell's energy needs and the availability of substrates. Definitely check out the video we just put out on this. Now let's walk this step by step. The ETC within ATP production is the sequence of events occurring across the inner mitochondrial membrane. That's what we call this guy right here, the IMM, inner mitochondrial membrane. Initially, complex 1, or NADH dehydrogenase, will receive electrons from NADH. This is going to initiate the chain. These electrons then move to CoQ, or ubiquinone, which is reduced to ubiquinol. Simultaneously, complex 1 is actively pumping proton into the inner membrane space, creating a proton gradient. Complex 2 will do a really similar thing, except with FADH2, which, if you'll remember, we made during the citric acid cycle, or TCA cycle. We'll see that complex 2 is doing really similar things, still contributing. See that complex 2 is passing its electrons along to complex 3, where complex 3 will again pump more hydrogens out, and then pass that same electron to cytochrome C, which won't pump hydrogens out. Cytochrome C will kind of shuttle back and forth to move the electrons between complex 3 and 4 here. And once it gets to 4, we're going to pump some more hydrogens out. Finally, all of these hydrogens are going to go through the turbine to make ADP create ATP. The final spot these electrons are going to go is to an oxygen molecule to form. Now let's do a quick practice question. Imagine a scenario where the cell's ability to produce NADH is compromised. How would this affect ATP production? Take a moment to think about this. Well, without NADH, the first electron donor in the chain, the entire process of electron transport and proton pumping would be way less efficient. Remember how many more NADH we made compared to FADH2 from glycolysis? This is going to lead to a significant drop in ATP synthesis, which will have widespread effects on the cell's ability to perform energy-dependent tasks, which is most of its tasks. Now let's do another super common question I see on the MCAT all of the time. Consider what would happen if the mitochondrial membrane became highly permeable to protons. Pause and think about this. Well, if protons could move freely across the membrane without passing through ATP synthase, well then the whole point of it is useless, right? The protons won't spin the turbine, they're just going to move across. And if they don't spin the turbine of ATP synthase up here, we're going to make no ATP. So just like in the first example problem, we are going to make much less ATP, which can cause big problems for the cell. And this is something people have done before. We've actually made weight loss drugs that disrupt the hydrogen gradient. And those drugs were discontinued because there was a high number of people that were dying from them. Today, we've journeyed through the cell to uncover secrets of ATP production. It's a complex but fascinating process, critical for life as we know it. Understanding these principles is not just important for the MCAT, but for your future medical career. Thank you so much for watching our video on ATP production, and I'll see you next time.